Okay, so good evening, student. Hope all of you are doing well. So, in the previous class, we have discussed about the function of mitochondria. Basically, mitochondria is a cellular organelle that helps in oxidative phosphorylation, or we can say that it helps in the process of respiration, due to which energy is released in our body. Now, it is also said to be as the powerhouse of cell. Now, so today we are going to discuss another cell organelle and we are going to cover the topic of cell. From tomorrow we are going to start another topic, that is the nucleic acid, the genetic material which is present inside our nucleus. Fine. So our next cell organelle is plastids. So first of all, J. Shimper was the scientist who proposed about the idea of plastid. There are three different types of plastids, that is leucoplast, which is white in color, chromoplasts, which are colorful, and chloroplast, which is green in color. Now basically the function of leucoplast is fat storage. It stores fat bodies inside the plant cells, or in those cells in which in which plastids are present. So basically these are the structure which are generally found in plant cell and some bacteria. So leucoplasts are white in color, they helps in fat storage. Chromoplasts are colorful, it gives other color to plant, like red color of carrot, like violet color of brinjal. So these are due to some kind of chromoplast. Basically in carotene named chromoplast is present, and in brinjal betanine named chromoplast is present. Due to the presence of betanine, brinjal are violet in color, and due to the presence of carotene, this is betanine, and this red color is due to the presence of carotene. These are the different types of chromoplast which is found inside the cell. So it is colorful, and the green color, this basically helps in photosynthesis, and this green color is found in chloroplasts, and these chloroplasts are found in leaves and green part of the plant. Likewise, all of you have seen the seedlings, or I can say the early plant, the stem is green in color because of the presence of chlorophyll in them. Now we have to discuss mainly about the chloroplast. Fine. So just like mitochondria, this is also a double membranous structure, means it is made up of two different membrane, the outer membrane, the inner membrane. Now let me draw the diagram of chloroplast first. So this is the chloroplast. It is its outer membrane which is continuous, while the inner membrane is in the form of folds, and these membranous folds look like the coin stack upon each other. Means ऐसा लगता है कि हमने सिक्के एक के ऊपर एक रखे. So these are the inner membrane which is discontinuous or in the form of folds. Now these stacked structure are said to be as grana, and surrounding these grana is a kind of liquid matrix. Surrounding these grana is a kind of liquid matrix inside chloroplast, and this liquid matrix is said to be as stroma. So just like mitochondria, chloroplast is also double membranous structure. Its outer membrane is continuous while inner membrane is folded. This chloroplast is also a semi-autonomous organelle. Just like mitochondria, it is also semi-autonomous organelle. Why? Organelle of prokaryotic origin. Again, this was the bacteria earlier during the evolutionary phase. These get stuck in heterotrophic cell and they started showing symbiosis with the heterotrophic cell. So these were the semi-autonomous organelle of prokaryotic origin. Prokaryotic origin means these organelle were bacteria earlier, and semi-autonomous means as these cellular organelle contain their own circular DNA. So this is the own circular DNA and its own ribosome in them, just like mitochondria. It can also perform half of its function by itself only. So this is ribosome and this is circular DNA. So chloroplasts are the double membranous structure. Its outer membrane is continuous, inner membrane is folded. It is a semi-autonomous organelle of prokaryotic origin. The outer membrane is continuous, but the inner membrane is in the form of folds, and these folds seems like the coin is stacking upon each other. This structure in chloroplast is known as grana. Surrounding this grana, there is a liquid matrix. There is a liquid matrix, and this liquid matrix is said to be as stroma. This chloroplast contain its own circular DNA and own 70S ribosome. That's why it is a semi autonomous organelle. Next thing is, what is the function of chloroplast? Chloroplast serves as kitchen of cell while synthesizing own food via photosynthesis. So by the process of photosynthesis, they can synthesize their own food. So plants do not have to depend for their food on any other organism. They can synthesize their own food. That's why plant is said to be as the autotrophic organism. Is that clear? So this is our next cell organelle. Next thing is nucleus. Nucleus. This structure was first of all discovered by Robert Brown, and this nucleus is said to be as the brain of cell. Likewise, our body have our own brain. Likewise, every cell has its own brain, and this brain is said to be as the nucleus. Now, let us suppose this is the nucleus of a cell. It contains some pore. Let us suppose this is the nucleus. This is the complete nucleus. This nucleus have small, these are also double membranous structure, just like chloroplast and mitochondria. So this is a double membranous structure. Its membrane shows the presence of some pore. This pore are known as nuclear pore, through which any molecule comes and goes inside or outside the nucleus. These are nuclear membrane. The fluid which is present inside the nucleus is said to be as, like the fluid which is present outside this nuclear membrane is enclosed by membrane, cell membrane, is said to be as cytoplasm. Same, the fluid which is enclosed by the nuclear membrane, it is known as nucleoplasm. Now, inside this nucleoplasm, we'll see a dense body of black color, and this dense body of black color is known as nucleolus. And same inside this nucleoplasm, we'll find a highly coiled thread-like structure. This is highly coiled thread-like structure. This highly coiled thread-like structure is said to be as chromatin fiber. And these chromatin fiber get condensed during cell division. Normally, when a cell do not divide, these structure looks like highly coiled fibers. But when a cell started cell division, these chromatin fiber get condensed and form a rod-like structure, and these rod-like structure are said to be as chromosome. Later on, when we start the class of nucleic acid, we'll define this chromosome again. Fine. So, nucleus was discovered by Robert Brown. It is double membranous structure. It is also said to be as the brain of cell. Nucleus contains different thing. It have membranous pores, and through these pores molecules enter or exit inside or outside the nucleus. It have nuclear membrane. It contains some fluid like structure which is said to be as nucleoplasm. It have a dense body, that is nucleolus, which helps in RNA synthesis. What is RNA? We will discuss it later when we start the chapter of what we can say, nucleic acid. And inside this nucleoplasm, we'll find highly coiled structure, and these highly coiled structure are said to be as chromatin fiber. When a cell is about to divide, these highly fibers structure get condensed and they'll form a rod-like structure, and these rod-like structure are said to be as chromosome, and these chromosomes serves as vehicle of inheritance. They'll carry gene from mother and father. That's why we call them as vehicle of inheritance. Fine. So these are the different cell organelle. Now let's talk about the cell coverings. So first of all, there were two cell covering. One is cell wall. So cell wall are the structure which is basically absent in animal cell. So these are the structure, we do not find them in animal cell, but cell wall are the outermost covering of number one, plants, number two, algae, number three, bacteria, number four, fungus, and number five, some protist, not all protist bear cell wall, some protist bears cell wall. So cell wall are dead structure, and these dead structure are freely permeable, means anything can pass from them. Most importantly, water can easily pass from them. Plus, these cell wall are made up of carbohydrates. So these cell walls are made up of carbohydrate or sugar. The only difference is that the type of sugar is different in different organisms. For a kind of example, if we talk about plants, algae, or some protist cell wall, they are mainly made up of a polysaccharide known as cellulose. Other than cellulose, other sugars are also present over there, but they are mainly made up of cellulose. Fine. If we talk about fungal cell wall, then fungal cell wall is made up of another sugar polysaccharide, that is chitin. While if we talk about bacterial cell wall, this bacterial cell wall is made up of another type of polysaccharide or any other type of sugar, peptidoglycans. And these peptidoglycans are basically two different sugar, NAG and NAM, N-acetyl glutamic acid and N-acetyl muramic acid, or N-acetyl glucosamine or N-acetyl muramic acid. So these are the structure which are said to be as cell wall. These are absent in animals. Due to this, we have a little bit of flexibility over our skin. These are the outermost covering of plants, algae, bacteria, fungi, and some protists. These are the dead structure and are freely permeable to water. And these are made up of carbohydrate or sugar. Plants, algae, or some protists, cell wall is mainly made up of cellulose, while fungal cell wall is made up of chitin, and bacterial cell wall is made up of another type of polysaccharide, that is peptidoglycan. And these peptidoglycans have different forms of sugar. One is N-acetyl glucosamine and another is N-acetyl muramic acid, that is NAG and NAM. Is that clear? Next thing, cell membrane. Cell membrane is a living structure and it is found in all cells. Whether it is prokaryotic, whether it is eukaryotic, doesn't matter. It is found in all cells. These cell membranes are selectively permeable. Means these can select a molecule whether this molecule can enter the cell or that molecule cannot enter the cell. That's why they keep on selecting the molecules and allow them to pass the cell on the basis of their structure, size, polarity. Fine. So these are selectively permeable. These are made up of lipid 85% and protein. So these are made up of lipids 85% and protein 15%. ठीक है, plus little carbohydrate. But the major composition is of protein and lipid. Plus, it is also known as cell membrane is also known as plasma lemma or plasma membrane. So we can call cell membrane as plasma lemma as or plasma membrane as well. So these are the another name for cell membrane. Now to understand the structure of plasma membrane, a model was proposed, that is fluid mosaic model by Singer and Nicholson in 1972, and they said protein icebergs in the sea of lipid. They observed the arrangement of plasma membrane is like there are lot of lipid molecule which is working as a sea. These are lipids. These are lipids. And on these lipids, there are submerged protein particles, which is working as an iceberg. These are the proteins particle and these are lipid. So these are serving as protein iceberg in the sea of lipid. If we talk about the thickness of plasma membrane, so it is nearly about 70 angstrom. 70 to 100 angstrom, mostly these are 70 angstrom. So that is all about the plasma membrane. So these are the membranes. So what we have studied today, we have discussed about plastids. ठीक है, plastid was discovered by J. Shimper. He proposed 3 different types of plastid are there: leucoplast, chromoplast, chloroplast. Leucoplasts are white in color, and the function of leucoplast is fat storage. Chromoplasts are colorful, and these give other color to the plant, like red color of carrot and violet color of brinjal is due to certain type of chromoplast present in those vegetables. Chloroplasts are green in color and helps in photosynthesis. These are found in the leaf and green part of the plant. Chloroplasts are double membranous. ठीक है, its outer membrane is continuous while inner membrane are in the form of pores, and these are semi-autonomous organelle of prokaryotic origin. These serves as a kitchen of cell by synthesizing own food by the process of photosynthesis. These folds are said to be as grana. These liquid matrix are said to be as stroma, and it contains its own 70S ribosome and own circular DNA. That's why we call it as semi-autonomous organelle. So that is our chloroplast. Then we have discussed about the nucleus. Nucleus was discovered by Robert Brown, and it is said to be as the brain of cell. And nucleus, if we see the structure of nucleus, there is a membrane known as nuclear membrane. These membrane contains certain pores. These pores are known as nuclear pore. Inside this nucleus, we found a liquid which is said to be as nucleoplasm. In this nucleoplasm, we'll found a structure, dense structure, which is called as nucleolus, and the function of nucleolus is RNA synthesis. And inside these nucleoplasm, we find a highly coiled structure which is chromatin fiber, which during the time of cell division get condensed and take the shape of chromosome, and these chromosomes serves as vehicle for inheritance. Now if we talk about the cell covering, we have two different types of covering. One is cell wall, another is cell membrane. Cell wall absent in an animal cell, and it's the outermost covering of plant, algae, bacteria, fungi, and some protists. These are the dead structure and freely permeable to water. These are made up of carbohydrate or sugar. In plants, algae, or some protein, these are made up of mainly cellulose. In fungus, these are made up of chitin. In bacteria, these are made up of peptidoglycan like NAG and NAM. Cell membrane are the living structure which is found in all cell. These are selectively permeable, and due to because they are selecting each and every molecule that you have to go inside that cell and you have to stay outside of the cell, we'll call them as gatekeeper of cell. We'll call them as gatekeeper of cell. These are made up of lipid 85% and protein 15% and of little carbohydrate. It is also known as plasma lemma or plasma membrane. Now to understand the structure of plasma membrane, many scientists like Overton was there, Gardner was there, Danieli Davison was there. Okay. Different scientists have proposed different model, but to understand the structure of plasma membrane, there is one widely accepted model. There is a fluid mosaic model which say protein iceberg in the sea of lipid. In the sea of lipid. This was given by Singer and Nicholson in 1972, and the thickness of plasma membrane is generally 70 angstrom, but it varies from 70 to 100 angstrom. So in our next class, we will discuss about either tissues or we will discuss about nucleic acid. Most probably we will start from tissue. Later on, when we start the topic of genetic, we will discuss nucleic acid in them. Fine. So thank you. Have a nice day.